So I want to start off by saying that in the spirit of reconciliation, the Civil Society of Australia acknowledges the traditional custodians of country and their connections to land, sea and community. We pay our respects to elders past and present. We extend our respect to all Aboriginal and Torres Strait people today, both in person and online. So welcome and thanks everyone to what is a very special event for the ACT branch of the SSA and the SSA, namely the inaugural lecture. My name is Francis, I'm the president for the SSA. Before I start, I do want to do a bit of housekeeping. So for those who are joining us online, please mute your audio to minimize feedback. We'll leave questions until the end of the talk. And if you're online and you want to ask a question at the end, please enter in the chat. And for those attending in person, you can obviously raise your hand and ask it. We will endeavour to try to repeat the question so that everyone online and in person can hear. Also note this meeting is actually being recorded. It will be made available, publicly available afterwards. Okay, so today on behalf of the ACT Branch Council, I'm proud to announce and introduce the Wilson Lecture, which is a new named biennial lecture that will occur at the end or as the last meeting of the SSA ACT Branch in even year cycles. The Wilson Lecture is named after the late Professor Sue Wilson and her immense contributions to the statistics and bioinformatics community in Canberra, both in Canberra, across Australia and globally. Sue has many accolades, and just to name a few of them. She was an elected member of the International Statistical Institute, a fellow of the American Statistical Association, a fellow of the Institute of Mathematical Statistics, and the inaugural senior fellow of the Australian Bioinformatics and Computational Biology Society. Now, I could speak more to her many accomplishments. I can also, but it does give me a great honor to say that Jonathan Wilson, Sue's son, is actually in the audience today, and he'll also be coming to dinner, so you can chat to him afterwards. I've also got a short message to play from Bruce Wilson, Sue's brother, and so I'm just going to try to play that now, so just bear with me for a sec. Share sound. Okay, so hopefully everybody can hear that okay.
Good evening. My name is Bruce Wilson, brother of Professor Sue Wilson. It was a wonderful surprise to receive an email from SSA ACT branch advising that they'd established the Wilson Lecture to honour my late sister, Professor Sue Wilson. Since we're unable to attend in person tonight, I've recorded this short video on behalf of my wife, Dr. Karen Byth, who's also a statistician, and myself. Sue's wide-ranging research interests progressed both theoretical and applied statistical knowledge, particularly in the areas of genetics, biostatistics, and bioinformatics. She worked tirelessly for the scientific community, both nationally and internationally, over many decades. Sue loved academic life and being involved in interdisciplinary collaborations. Many of her collaborators are lifelong friends. Sue was a true trailblazer for Australian women, particularly Australian women considering a career in statistics. She never gave up. She never gave in, except a rigorous scientific argument. After stoically coping with metastasizing cancer for 17 years, Sue was admitted to ICU in March 2020, just as COVID-19 entered our lives. Even then, Sue was reflecting on the similarities between COVID and previous pandemics that she had studied. Literally to her final hours, on the 16th of March 2020, just 3 days short of her 72nd birthday, Sue was never going to give up. It's wonderful to see Sue's important contribution recognised by the SSA. I'm sure that she would be thrilled that Gillian Heller will be giving the inaugural Wilson Lecture this evening. Best wishes, Bruce Wilson.
Okay. So, yeah. Thank you, Bruce, for a very lovely message, and thank you, Jonathan, for coming today. And it's a great honour for the SSA to be able to introduce a named lecture after Sue. So to present the inaugural Wilson Lecture for 2024, it is my great pleasure to introduce Professor Gillian Heller, who has graciously accepted this invitation. Gillian is currently the Professor of Biostatistics at the NHMRC Clinical Trial Centre, University of Sydney. After completing her studies at the University of Cape Town, Gillian migrated to Australia, where she has held academic positions at the University of New South Wales, Macquarie University, and the University of Sydney. She's the past chair of the Biostatistics Collaboration of Australia, past head of Department of Statistics at Macquarie University, where she currently also holds an honorary professorship. Gillian's research interests lie in distributional regression and biostatistics, in which she has published widely, including being the author of 4 books. She collaborates widely with colleagues in the UK and Europe, and in her position at the Clinical Trial Centre, Gillian works closely with clinicians and other medical professions in a consulting role. And I believe that's quite close and near and dear to Sue's heart, and something that Sue was also very passionate about. So today, Gillian will be presenting a talk around back to the future, or model what you measure. So let me bring that up. So just give me a sec, I'll bring that up. So, okay. Sorry, main controls. Okay, so hopefully everybody can see that online. All right, so please join me in welcoming Gillian Heller as the inaugural Wilson Lecturer.
Thank you for that introduction, Francis, and I'm very honoured to be the inaugural Wilson Lecture speaker. Sue was such a well-loved and much esteemed member of the statistical community, and it is indeed a big honour. I hope the title of the talk will become clear as I proceed, but I'm going to be talking about thinking about what we model, what as related to what we measure and what we want to get out of it. So let me get started. And I'll start with just getting a blank. Or the all models are wrong, but some models are used.
George
Box.
Yeah.
That's right. George Box. So we know that statistical models are abstractions of the real world. And in the physical sciences, those abstractions might be quite close to reality when the phenomenon under study is well understood. So I'm thinking, for example, of Newtonian physics, where the phenomena are really well understood and you would expect the model to be close to the real world, to reality. In other areas, such as social sciences, the abstraction may be more speculative. But in all cases, what we do is we observe data, we formulate a statistical model to describe the data generating process, which is a mathematical abstraction of the real process. Now, we have to think about why we're doing this. What is the purpose of the model? And if the purpose is prediction, then predictive ability is all that matters. So interpretability isn't all that important, and the model can be a black box, as it is in machine learning, and that's okay. Now, this is not what I'm talking about tonight. What I'm talking about is exploratory or confirmatory models, which we need to be interpretable, we need them to be useful, and we need them to be as simple as possible while achieving the first 2 goals. Now, I've used the word simple. What do we mean by a simple model? Well, what we have to do is distinguish between simplicity and mathematical convenience. They're not always the same thing. So we'll start off Stats 101, repeated observations of a binary outcome. So tossing a coin many times. And the most natural summary is relative frequency. And that is as close as we can get to the data and is easily interpreted as probability. Now, a commonly used summary is odds. Now, what are the odds? It's the relative frequencies of occurrences to non-occurrences. Now, why would you look at that? That's not all that intuitive. Well, it's mathematically convenient, which we'll see later. And because of that, it's everywhere in the analysis of binary and categorical data. I have to say, the odds are used in the gambling world, but that's the only area that I can think of where, besides statistics, where they're actually used. So relative frequency or probability is a the data. It's one that we understand naturally. The odds are complex, but mathematically. And I've just added this more French so that I don't get into trouble. For case control studies, we can't use probability, we can't use relative risk. We have to use the odds because that's what makes sense in that context. So we're forced to that. All right. So keeping on the track of binary outcomes, we think of the simplest and of modeling a binary outcome as a function of a binary predictor. So that can be a risk factor or an exposure or a treatment allocation. So here's a table that we're all familiar with. We have our predictor, which is 0, 1, yes or no, on or off, exposed or not exposed, and the event occurrence, yes or no, and then we have frequencies for each of the 4 combinations. And then we can write down the event rate for the predictor at level 0 and at level 1, so R0 and R1. So the event rates R0 and R1 are alternatively referred to as risks. Fortunately, those start with R, so we can do that. And then to quantify the discrepancy between the event rates, we can either use relative risk, which is the ratio of R1 to R0, or the difference, R1 minus R0. So those are both perfectly valid quantifications of the difference in event rates, the difference or ratio between the predictor at level 0 or 1. So if you would say the risk is doubled or there's a risk difference of 10%, those are concepts that are unlikely to be misinterpreted. And if we keep going with that, and if we extend the analysis to a binary outcome with multiple predictors, so multiple regression with the binary outcome, like so Y has the Bernoulli distribution with probability parameter P, and then some function of P is we get the picture on that. So if the G, that's our link function, if the link is the log, then log of P equals X beta, and then each of the beta gives you your relative risk. So that's a relative risk regression. And if we use the identity link, then beta J is the risk difference. So by varying the link function G, we very easily define regressions on the scale of relative risk and risk difference. So that all comes out very nicely. However, these regressions are infrequently used. We don't see them very much. Why is that? It's because the log and identity link functions don't guarantee the fitted probabilities in the 0-1 interval as we need them to be. We can overcome that, and so some work has been done by my colleague Ian Marshner and his PhD student, and they've really solved it. They've written some quite complex, they've devised some complex algorithms to do with constrained optimization, and they've really cracked that one, and they've got an R package log bin for relative risk regression. So it can be done. The mathematics and the computation wasn't that easy, but it has been achieved. However, what we know and mostly love is logistic regression. That's the go-to method for binary outcome. And so it's based on the logistic link function, and that gives you effects on the odds. So if your link is the logistic link, then each of the beta gives you your odds ratio. And the good news on that is that the logistic link function is the canonical link for the Bernoulli distribution. So it delivers your fitted probabilities in the 0-1 interval, as will the invoice of any sigmoid function. But the logistic is the one that's mathematically the easiest. Computations of MLEs is straightforward. And logistic regression is available in all statistical software packages, and it's the go-to method for binary outcome data. However, my contention is that the odds scale for regression effects is far less intuitive than relative risk. How do they compare? The relative risk and the odds ratio are approximately equal when R naught we think of as our baseline risk. It's the event rate when the predictor is at level 0. So, for example, if your event is, if you're thinking of a vaccination setup, your predictor 0 is unvaccinated and 1 is vaccinated, and your event is getting the disease or not getting the disease. Right, R naught is what we call the baseline prevalence or event rate. It's the disease rate for the unvaccinated population. Now, if we're looking at a rare disease, so R naught very low, and in fact, the odds ratio and the relative risk are about the same. You can show that easily mathematically. They just happen to be similar. So it doesn't really matter in that case which one you use. When your relative risk is greater than 1 and R naught is not close to 0, then your odds ratio is always greater than your relative risk, and increasingly so as R naught increases. So I've drawn a plot. This dotted line here is the line of equality. So I'm plotting the odds ratio against the relative risk for different values of R naught, your baseline rate. Now, for low R naught, 0.01, that line coincides with your line of equality. So there's no difference. It doesn't matter which one you use. But for high R naught, so 0.4, that's this blue line here, you can see that the odds ratio is very different from the relative risk, and increasingly so as your relative risk increases. And the red dot that I put here, that's the point, R naught equals 0.4, R1 0.8. And so that's a relative risk of 2, but an odds ratio of 6. Now, mathematically, they're saying the same thing, but intuitively, if I tell you that an odds ratio of 6, you're going to go away thinking that's 6 times the risk. Even if that's not quite accurate, what do you mean by risk? Is it probability or odds? Well, in this case, it's odds, but it's interpreted as probability. So that's a bit alarming. So while both measures indicate a substantial increase in risk, and qualitatively that lead to the same conclusion, meaning that you would reject the same hypothesis, an odds ratio of 6 is far more alarming than a risk ratio of 2. So in general, the use of logistic regression will lead to qualitatively the same conclusion risk regression. So I'm not saying that scientifically a whole lot of stuff that's been done has given wrong conclusions. That's not true. Qualitatively, the conclusions will be the same, but the quantification of the risk could be misleading it can be misleading if the increase in risk is important. So logistic regression is a mathematically and computationally convenient method, but it's a complex abstraction of the data. Now, I did a PubMed search just to see so PubMed is an archive of articles in the medical field. Right. And I did a search on firstly logistic regression, and the second search term I used was relative risk or log binomial regression, which it's also called. And look at this. Now notice the y-axis is on the log scale, and there's logistic regression going up at a huge rate, and log binomial relative risk regression is right down there. It's nowhere. It's so widespread use of logistic regression and very sparse use of relative risk regression. Now I'm going to introduce a data set to illustrate the concepts, and it's one that I've come across in my clinical work. There's an observational study, 366 patients who had head and neck cancer, and these patients were advanced. Their cancer had already spread, so their outcomes are typically very poor. The idea was to identify predictive risk factors for mortality. So this is the data that I had. The outcomes, firstly, 5-year all-cause mortality, and that was 49%. So almost 50% deaths after diagnosis. Well, not diagnosis, to start of the study. Time to death, a median of 3.88 years. Censoring rate, we'll just come back to this later. So 36% for overall survival. That means at the end of the study, then 36% were still alive. Whereas if we look at disease-specific survival, that's just something I'll look at later, 78%, so lower rate if you just consider mortality due to the disease, only 22% died. So those are the outcomes we will be considering. The risk factors, number of deposits, so they detect on scans deposits of the disease, of the diseased cells. So they count the number of deposits. They record the size of the largest deposit, so that's millimeters. And then this extranodal extension, or ENE, is a binary variable, which means that the cancer has spread through a lymph node into the tissues surrounding it. So the first thing I want to do is the simplest analysis, which is look at 5-year mortality as the outcome and ENE, a binary predictor. So there we have it. So for ENE, no, so the cancer hadn't spread through the lymph nodes. There was a 37% mortality, and for ENE, yes, 52%. So clearly, ENE has a harmful effect. That gives a relative risk of 1.4 and an odds ratio of 1.84. So the next statistical question is, well, is that a significant difference between the two ENE groups? And it doesn't matter which way you cut it. You can do a chi-square test, you can do a Z test for the log of the relative risk, you can do a Z test for the log of the odds ratio. They all tell you the same thing, that the difference between the two groups is significant. So no misleading there. And so qualitatively, the conclusion regarding the significance of association of ENE with 5-year mortality is the same. But if the magnitude of the effect matters, then the odds ratio implies an 84% increase in risk. However, that's understood. And the relative risk shows a 40% increase, which is less than harmful. Now, what I'm doing is going on, so keeping with the binary outcome, going on to do multiple regression with the other predictors. And so I did some model selection, and I found that the significant predictors were size of the largest deposit and log number of deposits. And so what I've got are the estimates on the left-hand side here for logistic regression, that's the odds ratio, and a 95% interval for a 1 millimeter. So that's a 1 millimeter increase in size, or log number, or an increase of 1. And then on the right-hand side, we've got the log binomial or relative risk regression. So what we see again is that an increase in size leads to an increase in the odds of mortality or the relative risk of mortality. So same story, but the odds are always greater. And same thing for log number. So more deposits means worse outcomes, higher mortality, but the odds ratio is giving us a higher number. So both predictors significant in both models, but the odds ratio is larger. Okay, so what I'm going to do now, so that's as far as I want to go with the binary outcomes. And so what I'm trying to show is that a method which is perfectly intuitive, doing a regression relative risk, or and you can also look at risk difference as the outcome, is doable and yet and give sensible answers and yet isn't much used. I'm going to move on to something a bit which is going to be a bit more controversial, and that's time to event outcomes. So survival. And the typical feature, as we know, with this sort of data is right censoring. So what I've done here is I've done a density estimate of the time to death. The blue density is of those that have died, so where we've observed time to death, and the pink ones are the ones who censored, so that's the time to end a follow-up. Now, that's not really valid. We can't really look at those times to end a follow-up, or they haven't died, so those times are going to be longer. But that's not really a valid comparison. What we should be looking at is the survival curve or Kaplan-Meier curve, and what we see is that the patients drop off pretty evenly through the life of the study. Now, how would we analyse this data? Now, what I'm asking you to do is just empty your head of what you would do and just think about it from first principles. We've observed the time to death. Sometimes we don't observe it because that time is censored, in which we observe time to last follow-up, and we know which ones they are. We've got a censoring indicator that tells us. What could we do? Well, we could do a regression for time to event, and we could write down a GLM type model. So it could look something like this, that the time to event given some covariates, and we need the CI is a censoring indicator. We know that's going to be relevant. It's got to be in there. That has some distribution with a mean and some sort of dispersion parameter. And we'll put a model on, well, I'm using a log link because obviously the location parameter has to be negative. Log of mu is some linear predictor. So D is some distribution with support on the positive real line. So maybe it's the Weigel, the gamma, the inverse Gaussian. You can write down a whole lot of continuous distributions on the positive real line. Mu is a location parameter, could be the mean or the median. Sigma is some scale or dispersion parameter, could be the variance or some other scale parameter. And the beta J's are additive effects on log mu. Now, likelihood maximization for a model like this is straightforward, as long as you can compute that distribution. The censoring issue makes estimation a little bit more complicated, but it's completely doable. So, I'm going to indulge in a little bit of history, a little bit of history lesson at this point. Now, I started in 1960, just because none of us were born before that. The world didn't end in as far as I need to go. Now, the linear model has been around forever, so we don't have to worry about when it first came into the literature. And we all know the linear model. The GLM, so Nelder and Wedderburn, seminal paper, 1972. And that really revolutionised what we could do in those days, 1970s. Well, they really came into use, it was published 72, really came into use in the early to mid 80s. And because of their clever, iteratively weighted least squares algorithm, they were doable with computing at the time, which was pretty primitive. I mean, a great big computer, which would be the size of this room, would have less computing power than my mobile phone. So that was very clever. Now, what came next? I'm going to introduce a whole lot more models, which is not comprehensive, not exhaustive. They're just my favourite model. So if your favourite model isn't here, don't be offended. It's just my view on it. Well, in the 1980s, Hastie and Tibshirani came up with the generalized additive model, or GAMS. And so, this curly E I'm using as notation for exponential family. So GLM's exponential family with parameter mu I, and then constant dispersion parameter sigma, and then you put a model on mu I. The GAMS keep that, but they allow smooth terms for the x's. And that was a big step forward. Now I've got some favourite models of mine, but as I say, there are lots of others. There was a lot of development around this time. This one here, and I've got some Australians, this was Murray Aitken. What he did was he took the normal model and put a model on sigma. So that was pretty revolutionary. And then this down here, another Australian, Gordon Smythe, took a GLM, the gamma model, but allowed a model dispersion parameter. So you can see the direction that things are going in. And then Rigby and Stassinopoulos in the UK came up with, these were called mean and dispersion additive models. These were still in the exponential family, but they allowed a model on mu and a model on phi, the dispersion parameter, and they allowed smooth terms for both predictors. Right, and there was lots more, but the important one in 2005, the same authors, Rigby and Stassinopoulos, published GAMLs, and this was a seminal paper. So GAMLs is generalized additive models for location, scale, and shape. What it is, so Y can have any distribution. This D is any distribution that's computable. The distribution can have any number of parameters, although usually it's up to 4. In theory, it can be anything, but you wouldn't really want more parameters. And then you can put models on some or all of the parameters, and you can have smooth terms. So that's a very, very general framework, which is still very much alive. So they've written the GAML software, and that's very much constantly being developed. I'll also just put in there quantile regression because I'm going to be talking later, but that's also important because if you think about GAMLs, that's modelling not just the mean distribution. And so we've been using the term distributional regression because of that feature. And quantile regression can also be classified as distributional regression. That was Roger Kuhnke in 1978, first published quantile regression. And he's very active and he's got R packages. So let's think back now to our survival models. How could we analyse our time to event T? Well, we could write down a GLM. We could say T comes from the exponential family, and we could put a model on mu. You'd have to incorporate the censoring, of course. So the response distribution would be the gamma or the invert Gaussian. It's limited to those in the exponential family. Or you could go for a GAMLs model, which is very general. So you could say your time to event is some distribution on the positive real line. And then you can put a model on mu and sigma. I've just written down a distribution with this. You can have one, but let's just keep it at 2. You can have a model for the location and the scale. Now I've left a blank in the middle there for a reason. And that is the so-called accelerated failure time models, which I think the problem with these models is their name. They just sound much more complicated than they are. It was a really unfortunate choice of name. What they are, they do a regression on log of time. So log of t is on the real line, and so once it's on the real line, it can take any distribution. So the error term has some distribution, 0, and which can be the normal, the logistic, and the Gumbel, which then translate to a corresponding distribution for time, and you can do the maths on that. So these are the possibilities. And so I've put AFT, when did it come about? I've put it there at 1980. In fact, I found our struggle. There isn't actually a seminal paper which introduced AFT. They first arose in the engineering world, and they're still popular there. And then they migrated over to mainstream statistics and the medical world. So I couldn't put my finger on a paper that's when AFT was introduced, but around the 1980 mark. So I've implemented all of those. So with my head and neck cancer data, I did an AFT, the accelerated failure time model. The Gumbel 01 model was chosen for the error distribution, which implies an exponential distribution for time to event. And then the predictors were the size of the largest deposit and log number of deposits, as they were for the binary outcome. And so that's your model. And then I did a Gamel's model, and what came out was the Weibull distribution was chosen with the same predictors for mu, size, and log number. But then we've also got the option of choosing a model for sigma, and log number of deposits was chosen for sigma. So that's your model there. T is Weibull, and then we've got a model for mu and a model for sigma. And there they are. Now, here are the AFT results, and here are the Gamel's results. And so the first two lines are the models for mu, and then the second is the model for sigma, which only exists for the Gamel. So firstly, notice that we're not comparing apples with apples, because in the AFT, mu is the median, and the Gamel's mu is the mean. So a little bit different, but actually the coefficients are remarkably similar. Minus 0.014, minus 0.012, and similarly for log number. So not too much difference. So for 1 millimeter increase in size of the largest deposit, the effect on the median for the AFT is 1.4%, and the effect on Gamel's, the effect on the mean is 1.2%. So 1.4% reduction in survival time. So a bigger deposit is harmful, and that reduces your survival. So a median and mean survival time are reduced, but you know, this is a difference that's great. Now, how do we interpret the effect of log number of number of deposits? For the AFT model, that's easy. We can do the same sort of computation. It would be a multiplicative effect because it's logged here, but we can do that computation for the AFT model. For the Gamel's model, we can't. If we said if the number of deposits is increased by 1, what is the effect on the mean? Well, we can't, because that also changes the variance. It also changes the shape. So you have to consider the effect of a change in covariate on the distribution as a whole, because the mean and the variance, the whole thing is changing. And so the way that we do it is by looking at the predicted PDF. And so you have to choose a value for the other covariates. So I fixed size at its mean, and then I've looked at the predicted PDFs for number of deposits 1, 2, 3, 4. And so for the Gamel's model, so for example, so this orange one is 1 deposit. And so we see there's less at the origin and more in the tail, so longer survival times. 1 deposit. And if you look at 4 deposits, there's more at the origin and less in the tail, so shorter survival times. So that's the story that's telling us. For the AFT model, we could just have interpreted the parameter, but I've done the same sort of graphic, And it's the same story, excepting, as we can see, we have far more flexibility. Well, this is a Weibull distribution, which has got an extra parameter, as opposed to the extra. Of course, we can get more shapes with the gamma than we can with the AFT. Now, another approach, I mentioned quantile regression. I'll just look at this quickly. For overall survival, your censoring rate is 36%. So by the end of the study, 36 were still alive. For the disease-specific survival, 78% were still alive. So that's a problem, because how are you going to estimate mean or median survival when you're only observing survival in 22% of your sample? You can't estimate something that you can't see. So all that you can do when you have a 78% censoring rate is estimate the lower tail of the distribution, because that's all you're seeing. And so we use censored quantile regression, which Hooker has published, and he's got a very nice R package that does it. So for overall survival, so what the package produces is for every quantile, you can tell it what range of quantiles you want to look at. For every quantile, this is basically between 0 and 1, it gives the blue is the estimate of the effective size, and then we've got the confidence band around it. So we can see different effective size. The effective size is greatest at about the 60% quantile, 60th percentile. For log number, it's greatest probably at about the 11%. And what I've superimposed in red is the estimate from the model we've just looked at, the Weibull model, and it's a little bit different. So that's quantile regression, where you can look at all of the quantiles. However, for disease-specific survival, it's just nonsensical to be looking at anything above certainly your 40th percentile. I mean, as we can see, it just goes crazy, and these are worthless. But it is useful, and so we wouldn't the Gammell's, this comes out of the Gammell's regression, gives us an estimate for the 50th percentile, but it's worthless. The estimates that are useful are the lower quantiles. And so this is, an advantage. Distributional regression, you can model the whole distribution, and quantile regression gives you this capability when all you've got is the lower tail of the distribution. So distributional regression models do a pretty good job of explaining time to event. However, we all know this isn't what we do. Right. So here I come back to my timeline, and surprise, the Cox model, 1972. And think about it, 1970, where were you in 1972? Two seminal papers that changed the course of statistical analysis, both published in the same year. That's quite amazing. It wouldn't have been appreciated at the time, but now we can look back and see that. So the Cox paper in 1972 set the course for analysis of survival data. And here's the model. It's not based, we're not doing a regression on time to event, we're doing a regression on the hazard function. And what is the hazard function? It's the instantaneous probability of an event at time t, conditional on survival to time t. Now, is that intuitive to you? It's not all that intuitive to me, and I can assure you it's not all that intuitive. I work with clinicians, and they throw about the term hazard ratio very easily. But if you ask them, what is it? They don't really know. And I can't tell very easily either. So it's a complex concept. So H naught is the baseline hazard function, so common to all subjects, and then it's modified by that e to the x beta to get the hazard function for a subject. So e to the beta j's are multiplicative effects on the hazard function. Now, there's no assumption regarding the distribution of time of event. So it's called a semi-parametric method. But the implicit assumption that the hazard function has the same shape for all subjects, and that's the term proportional hazards. And it's a go-to method for the analysis of survival outcomes. However, I'm saying that the hazard function is a complex abstract of the data. Now, I went back to PubMed and I wanted to see how much it's used. So again, the y-axis is on the log scale. I did a search on the terms proportional hazards regression or Cox regression. I also just threw in Kaplan-Meier because that's used a lot. That's, just your plot of the survival function. And then accelerated failure time or parametric survival. And here we get the results. Well, Cox regression and along with it Kaplan-Meier, are everywhere. They've taken quite the interpretation. They're what everybody uses. Very sparse use. Look at the numbers, 100 for parametric survival. So there you go. And it's interesting that graphic is so similar to the one logistics risk regression. It's quite striking. So you get a method that takes over, and these other ones that never quite get traction. So I computed the Cox model for my head and neck cancer data, and again, size and log number were significant. However, this p-value for the proportional hazards assumption tells us that the assumption is violated strongly for both covariates. So it's not really valid to use this model. So although both appear to be important risk factors, we can't actually estimate their effects using the proportional hazard model. Just to give you some insight into why. For proportional hazards, if the hazards are proportional, your Kaplan-Meier curves, if you graph them by your groups, they'll be parallel, they won't cross. And you can see for both size on the left and number on the right, they're crossing. So non-proportional hazards. So what have we found out about the effect? Let's just take size of largest deposit. A 1 millimeter increase in size, we saw before a 1.4% decrease in median survival time, a 1.2% decrease in mean survival time that came out of the Gables model. And I've put this in grey because we shouldn't really use it, but a 1.2% increase in the hazard rate. So again, it's telling the same story that a bigger tumour or deposit is bad for you, it's bad for your survival, but they're telling the story on a different scale. Now, I don't know how many of you have read this. It's very nice bedtime reading. In 1994, Nancy Reed interviewed, had a very wide-ranging interview with Sir David Cox. And it wasn't just about the Cox model, it was about his whole lifetime of work. And it's amazing. I'd really recommend reading it. But it's he said he'd been approached by quite a few people who said they were getting a certain kind of data, censored survival data with a lot of explanatory variables, and nobody knew what to do with it. And there seemed to be dissatisfaction with assuming an underlying exponential or Weibull distribution. And then it's quite funny if you go back and read the article. He had a bad case of the flu. And while he had the flu, he thought about this solution. He worked out the solution in his head, but he didn't write it down. And then when he recovered from the flu, he tried to remember and he couldn't remember it. But it took him quite a while, it took him a few months until he worked through it and then and found the solution. I have to say I've never had any insights when I've had the flu, but this is Sir David Cox. So he published this method, and the breakthrough was that it divided the likelihood into two parts: a part that involved your covariates and a part that involved your baseline hazard, and that enabled maximization of the part that involved the covariates only without estimating the baseline hazard. So that led to estimation which was feasible at the time. And again, think about the time it was, published in 72, probably getting to be late 70s, early 80s, when computers were as primitive as they were at that time. So being clever with algorithms, devising these clever methods was really important at that time. So by 1980, there were two competing regression models, the AFT model and the proportional hazards model, and the proportional hazards model completely eclipsed the AFT model in popularity and continues to do so. In Nature's list of most cited papers of all time in all fields, the Cox 1972 paper is ranked 24th, which is amazing. And in fact, I contend that that's an underestimate, because certainly in medical publications now, anybody analysing survival data will use the Cox model and they don't reference his paper. They either just use it without reference because it's so mainstream, or they'll reference a textbook. So that's an underestimate. So Cox himself said some surprising things. He said, when he asked how he felt about the cottage industry that's grown up around it, he said he didn't know. He said, in the light of some of the further results, I think I would normally want to tackle problems parametrically. So I would take the underlying hazard to be a Weigel or something. I can't see, not there. I'm not keen on non-parametric formulations usually. If you want to do things like prediction, it's much more convenient to do that parametrically. Another issue is the physical or substantive basis for the proportional hazards model. My point exactly. I think that's one of its weaknesses, that accelerated life models are in many ways more appealing because of their quite direct physical interpretation. So there it is, he said it, but by that stage, nobody was listening. So there we are. So statistical methods which are entrenched as the standard for analysis may not necessarily be based on conceptually simple abstractions of the data generating mechanism. They may have gained acceptance due to their mathematical or computational convenience, and the computational convenience would have been related to computational power at the time those methods were devised. And so while odds ratios and hazard ratios qualitatively deliver the same information as more intuitive quantities, such as relative risks and effects on the mean or median survival time, quantitatively, I think they're not well interpreted. Now, I don't want to give the impression that I'm dismissing the whole of the Cox model, because there's a huge knowledge base around that. So I want to talk a bit about survival data. It is structurally different from other sorts of outcomes because of the temporal nature, and because also the covariates can be of a temporal nature. So it is quite different. Now, regression modelling can be accomplished on different scales. It can be on the scale of the hazard function, and we've seen Cox regression and its many variants. It can be on the scale of time, t, that could be your AFT regression. It could be distributional regression, which could be GAMLs or other distributional regression models, or quantile regression, and you put a model on some function of the location parameter of the distribution of time. And you somehow cope with the fact that some observations will be censored. Another class of models, which I have to say I don't have any experience with, but I came across this when I was doing a reading for this talk. And those are models on the survival function. It just looks quite interesting, just another scale on which one can specify a model. So some function of the survival time, a survival function is a linear function of your predictors. So I think that's quite an interesting one, let's say, but I don't have any experience. So about Cox regression, as I say, I'm not dismissing it entirely. There is a very rich body of survival modelling based on the hazard function. And while the hazard function is not observable and perhaps less well understood than the survival function, it is informative of the course of the disease, if you can estimate it. And then proportional hazard regression has the ability to incorporate them each of time varying covariance and time varying coefficients, which distributional regression at this stage, I haven't seen any solution to that. So I don't know how you would do that. And I'll just say a violation of the proportional hazard assumption can sometimes be solved with a time stratified model. So there are ways of getting around that weakness. And I'll just say it is almost impossible to find a discussion of time to event without mention of the hazard function. It just pops up as soon as there's talk of survival data, the hazard function enters into the discussion. However, I feel that the AFT model the AFT model is a sadly neglected correlation of survival analysis. And yet I think it was ahead of its time. It was early on, before a lot of distributional regression. GLMs were only just getting known around then, but it was up there with those models. And it foreshadowed the development of the very rich framework of GAMLs. GAMLs and those distributional regression models have superseded it. You can do so much more. But the AFT was the forerunner, and yet it's been neglected. So as far as possible, what I would recommend then is to model what you measure. If you measure a time, then model the time. If you measure occurrence of an event, model its frequency. That will be the closest to the data that you get, and prioritize interpretability over mathematical convenience or computational convenience. So that's all I've got to say. I've just got some references there to log binomial regression. I've written up this talk in that second reference there. And at the bottom, I couldn't resist sticking in a reference to a text which I published with colleagues in the UK and Europe on the latest developments in GAMLs. Okay, thank you.
Very interesting. Anybody have any questions or comments they would like to ask? Castle.
That was amazing. Thank you, Gillian. One of the things that intrigued me was choosing the link function. So it seems like there's so many different processes involved in modeling, and you talked about a few of them. Why do you think changing the link function doesn't happen more often? How do you choose the question?
Sorry, just to repeat the question.
Should I? Yes.
Yeah.
So the question was, changing the link function is another aspect of model building. And why isn't that? We don't see much of that in model selection. Why isn't there more attention paid to that? I agree. Yes, that is because changing the link function changes the scale on which you're measuring. And yes, I agree with you. I believe there's not enough attention paid to that. Usually the link chosen is just one that's convenient, and usually a range preserving link is just chosen. So for example, if you have a non-negative parameter such as a mean of a non-negative quantity, then the log link is used. And there's the questioning of that, and it could be. So yes, I agree with you. Choosing the link function would receive more attention than it does.
Thank you. Any other comments or questions? Is there anything online in the chat?
Yeah, there's a question from online. So James Chamberlain, thanks for a great talk. What is your opinion on flexible parametric survival analysis?
Should I repeat that?
Yep.
Oh, no, it's on the chat.
Oh, you got that?
Yeah. So you just chat.
Oh,
Yeah.
oh.
Yeah, just get back up to the top.
Can you do it?
Yeah, yeah. So.
I don't know. Flexible parametric survival analysis. I'm not sure what. Do you mean distributional? No, I don't know what you mean by that. Sorry, James.
Yeah. Yeah, it's a method where you model the hazard function, the baseline hazard, as well as, and you can do non-proportional hazards and stuff with it. So it's sort of partway between Cox regression and some of the other methods, I think.
Yeah, okay. I haven't met that. But yep, sounds like there are lots of variants on generalizing Cox regression. So yeah, that could be a valid approach. Thanks.
Any other comments or questions? Ben?
Thanks for the talk. I was just wondering if you could talk a bit more about why you think the interpretation of some methods of analysis are harder than others, and if that's just from like a verbal way of explaining them, and how maybe data visualization can actually assist in the interpretation of some of the harder to intuit methods of analysis.
Yeah, okay. So the question is why I'm saying that certain summaries of the data are not so intuitive, and whether visualization would help in the process of understanding them better. I guess to me it's just a very personal view. I just find the odds probability easier to understand than an odds. So some people, and I mentioned at the beginning, in the gambling industry, the odds are used. So presumably those people understand the odds very well. But yes, we could. I'm trying to think if you were expressing results on the scale of the odds, how you could visualize. I'm not sure about that. Or a hazard ratio. Not sure that visualization would help. But yeah, some of my colleagues don't agree with me. I've had reactions saying, I understand the odds as much as probability. Some people think the hazard function is an easy thing to understand. And for statisticians that may be, but I've come across articles in the medical field which I'll complain about the fact that every binary outcome is analysed as a logistic regression. And effects on the odds just are not interpreted in the right way. So yeah, but my answer is it's a personal view, and maybe you understand the odds better than probability. That's fine. Yeah.
Cool, thanks.
Maybe something technical. So what fitting procedure is used for gamma-SS? And has any theory been done for gamma-SS? Yeah.
Okay, so the question, what fitting
Procedures
procedures, and yes, so read the book.
Yeah, absolutely.
Yeah, no. It's restricted maximum likelihood. Penalized It's penalised maximum likelihood, penalised for smooth terms and random effects.
Yeah.
That's basically a maximum likelihood approach. There is also a Bayesian approach. So there's a software package, GAMLs, which is Bayesian GAMLs, and which is written by some of my co-authors. And so that's all out there as well. And yeah, the theory is all there.
So people have done an asymptotic theory for the main parameters of the model.
Well, for the likelihood approach, you've got the likelihood or all of the asymptotic properties of NLPs.
Okay.
Yep. For Bayesian, I'm not sure what the asymptotics are.
There's another question.
There's your question in the chat.
Okay. Have you looked at? Yeah, I looked for that data set. I looked at the Weigel distribution. I didn't examine the exponential, but I looked at the residual plots for that Weigel fit, and yes, it fitted very nicely. Yeah.
I guess coming back to sort of the communication question, what is your comment on, I guess, in the education space? Would you suggest sort of undergraduate degrees sort of incorporate these kinds of models more than Cox model logistic? Or is there going to be a lag?
I have great, can I repeat that?
Yeah, sure. Thank you.
So what Francis is asking, what about education? Should these more intuitive be incorporated in teaching? I'm wrestling with that one because we can't not teach the Cox model. We can't send graduates out there who are ignorant of the Cox model because they're going to be asked to do it. But I have a problem with that. So I'm wrestling with that one at the moment because I teach a unit. Well, half of it is GLMs, but half of it is survival. And we teach the Cox model and we mention parametric models in one small section. And I want to change that balance, but I don't think that the parametric part of it can overtake. We have to, just because they use that much, we have to teach the Cox model. And it's not wrong. It's not wrong.
Any other final questions for Gillian? Next.
So I think in principle, you could like experimentally determine whether non-experts are better able to correctly interpret these like these alternative methods. Like, is anyone trying to do that systematically?
Yeah, okay. The question was experimentally, you could do studies on subject to determine how they're understanding the different concepts. Has anybody done that? I'm not aware. Yes, somebody could do that. Yeah.
Sorry, there's two questions.
Very often, sure. I just want to add on that because I've used similar model in one of my projects in the past, and that's for government. And then we're talking about odd ratios and things like that. People just seem to like what's odd, and they want to be able to understand that a little bit clearly. So what I did do at the end is almost translate all the odds into something that's easy for them to understand.
Yes.
So I can see where this is coming from because in my line of work, it's important for them to understand the ratios and what odds means, etc. And even within the internal team, it was hard to identify exactly what that means and how to interpret that. So just kind of comments, just based on my experience using in that one project, it was hard to use that term, and we had to not use that term and use something else to explain the outputs.
Yeah.
So,
I mean, there is a one-to-one relationship between odds and relative risk, so you can always just do that translation.
Yeah.
Yeah, that's very fun because I remember doing something similar like that once. I just could not interpret logistic regressions. I found it a bit hard, even though I understood what they are and how they work, but I just couldn't seem to figure out how to interpret these logistic regressions. Anyway, I digress. My question is, have they ever used these survival analysis stuff, like the Cox Proportional Hazard Model, have they been used outside of survival analysis?
Okay, so the question is, have the survival models been used outside survival analysis? I'm not aware of that. So you mean it's another situation where you had a non-negative quantity that was censored?
Yeah, like that too.
No, I'm not aware of that. It's always it's always time to an event.
Great, final comment from Cass.
Oh yeah, well, the Cox model isn't used in spatial statistics, my knowledge, but the Kaplan-Meier way of estimating survival rates is relevant to spatial statistical methods. So
Okay.
that's because if you have an observation window that acts like the censoring, because you can't see what's outside the observation window, and it has so distance to items in a space is censored because you can only see the distance to items that are inside the observation window.
Yeah, I've never heard of that. Yeah.
Yeah, it's pretty cool.
Okay.
And actually, maybe the Cox process, and maybe the Cox model is used in spatial statistics. Actually, I don't know it well enough to say, but I haven't seen it.
Okay, so I think we should pretty much wrap it up here. So thank you, Gillian, again for a very interesting talk.